Dear students, welcome back. Let's discuss about the meiosis cell division today. Last class we finished about the mitosis. So we have a question here again for the meiosis. Let's see. Which of the following best describes the stage at which the primary oocyte is arrested during oogenesis before puberty? So they're asking you about the stage, stage at which the primary oocyte is arrested during oogenesis before puberty. So let's see here a little bit about the oogenesis, little overview. The details we will discuss in the gametogenesis. So how does the oogenesis occurs? This is the developing embryo, or you can remember like this also. If you remember, we have the embryoblast in the middle, right? In this embryoblast we have the hypoblast and the epiblast, right, towards the amniotic cavity epiblast. So by this, what is this one? The epiblast. The epiblastic cells, they begin to develop the primordial germ cells by the second week of IUL. Which week? By the second week of IUL. These primordial germ cells, which begins to develop from the second week of IUL, they begin to migrate to the yolk sac. And they reach to the yolk sac by the third week of IUL. And these PGCs, primordial germ cells, they begin to migrate to the gonads, migrate to the developing gonads by the fourth week. And these primordial germ cells, they reach to the gonads by the fifth week. So after the fifth week, these primordial germ cells, these PGCs, they begins to undergo mitosis. So they divide continuously till the third month. Then after third month, some of these primordial germ cells, when they begin to divide, they undergoes mitosis. They are called oogonia. What are they called? Oogonia. The primordial germ cells, when they begins to undergoes mitosis, they are called what? Oogonia. They continuously divide until the third week. So once they divide until the third week, some of these oogonia, they begins to undergoes meiosis. Some continues to undergoes mitosis. They divide continuously till before birth. Right? So in the middle, before birth, at about 5th month of IUL, this number of oogonia becomes about 7 million. How much? 7 million. But some of these, they undergoes death from 5th month to the 9th month. So that at the time of birth or just before birth, these oogonia, they remain about 700 to 800,000 only. See, from 7 million they reach, the oogonia reach to 7 millions by the 5th month, and they continuously divide till before birth. And till before birth, some of the cells, even though they divide, some of these cells, they undergoes degeneration, and they remain how much? Only 700 to 800,000. So some of the cells already undergoes meiosis at third month. They begin to go to the meiosis. Some rest of the cells till before birth, some of them, they undergoes meiosis here, some of them undergoes meiosis here, some of them undergoes meiosis here. So at the different weeks, they undergoes meiosis. The first meiosis, they begins by which? Third month. Rest of the other oogonias, they also continuously undergoes what? Meiosis. When they undergoes meiosis, they begins to undergo the meiosis, they are called what? They are called the primary oocytes. So these primary oocytes, when they undergoes the meiosis 1, these primary oocytes, when they begin to go to the meiosis 1, they get arrested in the meiosis 1 prophase, or you can say prophase 1, meiosis 1 prophase 1 in the diplotin stage. Which stage? Diplotin stage. Meiosis 1, prophase 1 diplotin stage, right? So this arrest in the meiosis 1 prophase 1 diplotin stage, it is because of the release of OMI, oocyte maturation inhibition factors. It is released by the granulosa cells. Hope you understand, right? So that they get arrested in the primary oocyte, in which stage? Meiosis 1 prophase 1 diplotin stage, and they will remain arrested until the puberty. And once the puberty arrives, these cells, they continues their the meiosis, and then they complete the meiosis 1, they becomes a secondary oocyte. The secondary oocyte should complete the meiosis 2 once it undergoes the fertilization, right? So hope you understand. So what are the stages? So the question they will ask you from here: the primordial germ cells, they originate from epiblast, and these primordial germ cells, they begin at which week? Second week. And from the second week, they begins to develop as the primordial germ cells, and they begin to migrate to the yolk sac. They reach to the yolk sac by third week, and by the fourth week, they begins to migrate to the gonads, developing gonads, and they reach to the gonads by fifth week. And by the fifth week, the primordial germ cells, they begin to undergo mitosis. They continuously undergoes mitosis, and by the third month, some of these primordial germ cells, they undergoes meiosis. Once they undergoes mitosis, we call them as oogonia. Primordial germ cells, when they undergoes mitosis, we call them oogonia. Some of these oogonia, by the third month, they begin to undergoes meiosis 1. But the rest of the others, they continues to mitosis. They continue mitosis till before birth. The number of the primary, the number of the oogonia, they reach to how much? Maximum 7 million by fifth month. But by the fifth month to ninth month of the IUL, some of these oogonia will degenerate. And just before the birth, how many numbers of these oogonia remain? They will remain only how much? 700 to 800,000. All of these 700 to 800,000, they enters to meiosis 1. That means before birth, how many primary oocytes do we have? We have about 700 to 800,000 primary oocytes we have before birth. And all of these primary oocytes, they arrested in which stage? The meiosis 1 prophase 1 diplotin stage. So hope you understand. And what is the factors which makes them arrest in this stage? This is the OMI, oocyte maturation inhibitor. This is released by the granulosa cell. So multiple questions we can create from here. OMI is released from, and what is the factor which arrests the primary oocytes in the meiosis 1? That is the OMI, right? And another one, in which stage the primary PGC begins to form. Another question, in which week the PGC begins to migrate to the yolk sac. Another question, in which week this the PGCs, they begin to migrate to the gonads. What is the maximum number of the oogonia forms? In which month the maximum number of oogonia we can count, right? So these are multiple questions you can get from this the explanations here. So what is the answer here? So they get arrested until the puberty, in which stage? That is our the prophase 1 of the meiosis 1. Prophase 1 of the meiosis 1, and this is in which stage? The diplotin stage. Hope you understand, right? So this is the answer. Hope you understand. So yeah, this is the answer we have here. So let's see the meiosis. What is meiosis? What is meiosis? Meiosis is a reductional cell division, right? Meiosis is a reductional cell division. So what happens in the meiosis? The meiosis, you have the mother cell 2n. It will be divided into the n two daughter cells. That means these are the haploid daughter cells. These haploid daughter cells further will divide into n and n. This comes with n and this comes with, and that means one mother cell will divide into the 4 daughter cells. Mother cell is diploid. That means the number of chromosomes are 2 pairs. And we can say a pair of chromosomes. That means the paternals and the maternals. So you know that we have total 46 number of chromosomes, right? And here the daughter cells are haploid. haploid daughter cells. Total how many daughter cells we created? We created the total 4 haploid daughter cells. So look at here, this 2n is mother diploid. It is divided into what? 2 daughter cells, they are haploid. That means the number of chromosomes here is reduced. This is our meiosis 1. So number of chromosomes reduced from 2n to n in the meiosis 1. So that meiosis 1 is what? Is a reductional division. Reductional division, right? And then below that we have what? The number of chromosomes same here, and here also we become n. That means it is equational division. The meiosis 2 is what? Equation. Equation means the number of chromosomes remain same from this n to this n, right? So that means the meiosis 2 is what? Equational division. That means the number of chromosomes do not reduce, right? But in the meiosis 1, from the mother cells to the daughter cells, what happens? The chromosome numbers reduce. That's why this is what? the reductional division. Meiosis 1 is reductional, meiosis 2 is what? Equational division. Another point here, the meiosis undergoes in the gametes. They undergoes in the gametes. That means they forms the gamete, formation of gametes occurs. That means they undergoes in the germinal cells. They undergoes in the germinal cells. That means germ cells. These germ cells forms what? Gametes. Hope you understand, right? And yes, we understand. The another point is what? Because of the meiosis, it creates the genetic variations. Genetic variations because of the crossing overs. Crossing overs. We will discuss about the crossing overs. The another point about the meiosis. The meiosis produces the aneuploid gametes. It produces what? Aneuploid gametes. Aneuploid means euploid is here. Euploid means equal number of chromosomes. Equal number of chromosomes. Aneuploid means not the equal number of chromosomes. That means mother cell has 2n, daughter cell has only n. That means aneuploid, not euploid. Euploid means what? Equal, normal. Aneuploid means what? Unequal. So that the meiosis creates what? Aneuploid the cells, aneuploid daughter cells, and it occurs in the germinal cells we discussed already, right? The germinal cells are what? The ovaries and the testes. In the ovaries they will create what? The ova. In the testes they will creates the sperm, right? The gametes. These are the gametes. Hope you understand. And it occurs in the 2 stages that we discussed already, that is the meiosis 1 and the meiosis 2. Meiosis 1, I said already that meiosis 1 is what? The reductional division. And meiosis 2 is what? Equational division, because I show you already the examples here. 2n becomes n and n. This occurs in what? Meiosis 1. That's why the number reduced to half. That's why we call this as what? The reductional division. But these are in the meiosis 2, it becomes n and the same, it remains n and it remains the same. So this one we call this as what? Meiosis 2. Hope you understand, right? So this is what? Equational division. So it starts with diploid and it ends with what? Haploid. That means starts with diploid and it ends with haploid. Haploid, hope you understand. Yes, so that this please remember for the exam. So interphase of the meiosis, same like as mitosis, we have discussed about the interphase, but little bit difference over here. The interphase stages has no differences. What are those the stages in the interphase? If you remember, that was what? That was our G1, that was our S, that was our G2, that was our M, right? So in this G, some of them they will enter into what? G0 phase as well, we discussed before. Yes or no? So these interphase are the same for the mitosis and meiosis, but the difference is what? In the mitosis undergoes interphase. This interphase is followed by mitosis. This mitosis 1 cycle. But in case of meiosis, what happens? They have the interphase one interphase complete, there will be meiosis 1 and meiosis 2. That means one interphase followed by 2 cycles of meiosis, but one interphase followed by only 1 cycles of mitosis. That is the differences. The other changes are the same as like in the G1, S, G2, what were the changes during the mitosis we discussed earlier in the phases of the cell cycles. It is the same as like there we discussed earlier. And the difference is what? Mitosis, one interphase followed by 1 mitosis, but in the meiosis what happens? 1 interphase followed by 2 meiosis. Once again, mitosis, 1 interphase followed by 1 mitosis, meiosis, 1 interphase followed by 2 meiosis, right? 2 cycles, meiosis 1 and meiosis 2. Hope you understand. So there's the meiosis in the interphase we discussed already here. So once again you note it. What are those? G1, S, G2, M. In the middle, G1 may enters to the G0 phase as well, right? So this one you remember, okay? And after that, let's go to the M phase. M phase has how many phases we said? How many stages? 2 stages, meiosis 1 and meiosis 2. And these meiosis 1 and meiosis 2 we divide like this, PMAT. What are those PMAT? If you remember in the same like in the mitosis, P for what? Prophase. But here prophase 1 and prophase 2 because 2 cycles of meiosis, right? Similarly, metaphase also 1 and metaphase 2 because 2 cycles. Prophase, metaphase, A for what? Anaphase also 1 and anaphase 2, right? And another one, prophase, metaphase, anaphase, telophase, right? Telophase also 1 and telophase 2, right? So that we have that prophase 1, metaphase 1, anaphase 1, telophase 1. This is for the meiosis 1, right? And again prophase 2, metaphase 2, anaphase 2, telophase 2. It will make what? Meiosis 2. Hope you understand, right? So this is the divisions. And let's discuss some points over here about these stages, okay? So let's see here. So meiosis 1 is forming what? 2 haploid cells. 1 diploid cell we have, and this 1 diploid cell is creating what? 2 haploid cells. That's why we call this as what? The equational division that we said already. And what the meiosis 2? Meiosis 2. The meiosis 2 has, see, we have 2 haploid cells. These 2 haploid cells will form N, and here also N, and 2 haploid cells will form 4 haploid cells. 4 haploid cells. So this is we called as what? Equational division, right? We the number of chromosomes, they do not decrease, right, in the daughter cell. Same like N and N we have, but here 2N is reduced to half. That's why we call this as the equational division. Another point you have to understand here, the prophase 1, we said meiosis creates the genetic diversity, right? So here prophase 1 is responsible for creation of genetic variation because crossing over occurs in the prophase 1. Because of what? Because of crossing over. Because of this crossing over, what do they have? They have the exchange of the the genetic materials. We will come into the details of this prophase 1. So another one, the metaphase 1. This metaphase 1, as same like in the mitosis, it creates the the equatorial plane. The chromosomes are arranged in the equatorial plane, but the differences in the mitosis and meiosis is what? In the mitosis, this equatorial plane is formed by the single number of chromosomes, but in the meiosis, the metaphase 1, it is formed by the the pairs of homologous chromosomes like this. So it will form the 2 layers in the metaphyseal plane. The metaphyseal plate is formed by the 2 layers of homologous chromosomes, so that half moves up, half moves down. So that exactly the reduction, after the metaphase 1, the reduction occurs in the anaphase 1. anaphase 1. So these half chromosomes, the half number of chromosomes moves to one side, half number of chromosomes moves to another side. That's why it creates a reduction in the anaphase 1, right? So that what happens here, during the anaphase 1, if unequal number of chromosomes moves to the opposite side, it creates the non-disjunction. So the non-disjunction is produced in the anaphase 1 or anaphase 2. Because the number of chromosomes divides into the two folds during the anaphase. That's why non-disjunctions occurs. What is that non-disjunctions means? Non-disjunctions means once we form the daughter cells, we wrote here n number of chromosomes. Sometimes more number of chromosomes moves to the one daughter cell and less number of chromosomes moves to the another daughter cells. That creates the unequal number of chromosomes in the two daughter cells. This condition is called what? The non-disjunctions. Hope you understand, right? So now let's go to the prophase 1. So prophase 1 we further divide into the 5 stages. How many stages? 5 stages. And let's learn the mnemonics. Little Jara, please dance daily. L for liptotin stage. L for liptotin stage. Jet, Jara, zygotin stage, right? Then P for what? Liptotin, zygotin, pachitin stage. Another one D for what? Diplotin stage. And another D is over here. That D is for what? Diakinesis. So hope you understand the stages of the sub-stages of the prophase 1. So what are the sub-stages? That is liptotin, zygotin, pachitin, diplotin and diakinesis. Little Jara, please dance daily. So please remember like this, okay? So now let's go to the prophase 1. This one we discussed already. The resulting daughter cells has what? Half number of chromosomes. Yes or no? Half number of chromosomes. But the DNA material is constant. The genetic material is constant. So the number of chromosomes becomes half. You can see over here. We have the 4 chromosomes. Finally, in the daughter cells, we have what? 2 chromosomes only. This is the result. Now let's go to the meiosis 1. In the meiosis 1, we will discuss about the prophase 1. In the meiosis 1, we discussed already. It is what? Reduction. So what happens? One mother diploid daughter cell will divide into 2 haploid daughter cells. Now let's go to the prophase 1. It is same like the prophase 1. It is same like that which happens in the the mitosis prophase 1. What happens in the prophase 1? The chromosomes condensed, and the chromosomes becomes short. That means the chromatins begins to shortens, right? And the homologous chromosomes, they begins to align together. Homologous chromosomes begins to align together. Similarly, the nuclear membrane degenerates. Begins to degenerate in the early stage. In the later stage, it degenerates. Similarly, what happens? The Golgi body and what happens? The endoplasmic reticulums, they also degenerate. Similarly, what happens? The centromere begins to move to the two opposite pole. It's the same like in the the mitosis, but some changes occurs here in the meiosis prophase 1. What are those changes? That means the synapses formation occurs, and then the crossing over occurs. What are those? We will discuss in the next slide. Look at here. So what are those? Synapses and the crossing over formation. Okay, now let's go to the stages of the prophase 1. That is what? Liptotin. In the liptotin stage, what happens? The chromosomes, they appears as a slender thread. The first point. Second point, yes, each chromosome consists of what? two chromatids, because these chromatids, they come from already from the S phase. Do you remember? Synthetic phase of the cell cycle. One chromosome divides into two chromatids, right? Because of the copy of the DNA. So in the zygote stage, what happens? Zygote stage, the pairing of the homologous chromosome occurs. The homologous chromosomes, they begins to pairs. The homologous chromosomes, they begins to pairs like this. This occurs in the zygote stage. And in the Pachytene stage, what happens? Homologous chromosomes means the paternal chromosomes and the maternal chromosomes, right? Paternal and maternal chromosomes, they comes and they align together. In the Pachytene stage, let's go. In the Pachytene stage, what happens? This, the homologous chromosomes here, can you see here? Two chromatids, right? This in the middle, in the centrosome, these two chromatids, they are attached together by the protein, right? The cohesin protein. And in the Pachytene stage, what happens? These four chromatids, they become visibles. The four chromatids, they becomes visible. In which stage? In the Pachytene stage. Before the Pachytene stage, in the zygote stage, they just undergoes pairing. These chromatids may not be visible clearly, but they become clearly visible during the Pachytene stage, right? Now in the diplotin stage, what happens? The chiasmata formation occurs. What happens here? The chiasmata, chiasmata, the chiasmata formation occurs. What is that chiasmata formation? Chiasmata formation means, look at here, we have the chromatids, another chromosome, homologous chromosome is here, right? So the chiasmata formation means these two chromosomes, this side and this side, they undergoes the pairings. They undergoes the pairings, and because of the pairing, they undergoes the exchanging of their what is this one? The genetic materials. They exchange their genetic materials. So because of this changing in the genetic materials, they create the variation. That is what chiasmata formation. Chiasmata formation means the pairing together, visible contact of the crossing overs. This is called what? Chiasmata. And what is the crossing overs here? The crossing overs also occurs. Crossing overs means exchanging of their genetic materials. They will exchange their genetic materials in between these two chromatids. That is called what? Crossing over. And these exchanging of the the genetic materials, it is visible. This physically visible the portion which looks like the X. It looks like what? X. So this the physically visible X linking of these chromatids, this is called what? Chiasmata formation. And exchanging of the genetic materials is called what? Crossing overs. And this pairing of these two homologous chromosomes, this is called synapsis formation. What is this? Synapsis formation. So that synapsis occurs during what? Synapsis occurs in the zygote, and during the diplotin, what happens? This synapsis, they undergoes the crossing overs, and because of the crossing overs, they exchange the genetic materials, and this visible crossing over is called what? The chiasmata formation. This is the visible crossing over is chiasmata formation, and this chiasmata becomes more visible during the diakinesis stage. Chiasmata becomes more visible during the diakinesis stage. Why they become more visible? Because this chromosome, it begins to move to one side, the another chromosome, it begins to move to another side. If you see, okay, so if they begin to move to this opposite side, where was the crossing over? Crossing over was here. Genetic information exchange was here. So when they begin to move to two opposite sides, they begins to pull to opposite sides, so that it becomes more clearly visible X. Look at here. It is going to become X. It looks like a X, so that this X, it becomes more evident during what? The dikinesis stage. This is the question for you exam, right? So the chiasmata becomes more visible during the X stage, right? So the synapsis occurs in which stage? Synapsis occurs during the zygote stage. What is synapsis? Synapsis means homologous pairing. Homologous pairing. Homologous means the same number of, we have the chromosome numbers 1 to 23. 1 to 23 coming from father, 1 to 23 coming from mother. So the pairing of one number paternal chromosome with one number maternal chromosome, this pairing is what? The synapsis. So this synapsis occurs in which stage? The zygote stage. And in the pachytene stage, the same homologous pairings, there, the tetravalent, the sister chromatids, they becomes more visible. During the diploid stage, in the same pairing, they undergoes what? The crossing over and chiasmata formation occurs, right? So the crossing over means genetic materials exchange. Chiasmata formation means the same genetic exchange, if it is visible, physically visible, this is called what? Chiasmata formation. So you can see the pictures over here. Leptotin, in the zygote stage, the homologous pairing occurs, that is what? The synapsis formation. And during the pachytene stage, see over here, we have the crossing over going on. During the diploid stage, they begins to they exchange the materials, right? So in the pachytene stage, the tetrad becomes more visible, chromatids become more visible. During the diploid stage, what happens? They begin to exchange the materials. That exchange of the materials is called what? The crossing overs. And then the physically visible, that is chiasmata formations. The chiasmata becomes more visible during the dikinesis. Let's go to the metaphase 1. Same like the nuclear membrane completely degenerates. Same like the mitosis, completely degenerates, right? The chromosomes becomes very short and very thick, very short and very thick. So that this is the best stage. This is the best stage for chromosomal study. The spindle fibers, the ester formation already begin in the prophase stage. Ester formation begins already in the which stage? Prophase stage. Ester formation means already the formation of the microtubules by the centromere, right? So that was our what? The ester formation. From the ester formation, what happens? The spindle fibers formation begins. Spindle fibers begins, and these spindle fibers, they will go and attach with what? Centrosome, like this, right? So the formation of spindles completes, and the homologous, the chromosomes, both sides, like we have the homologous chromosomes, it is same like that of the mitosis. If you remember, the spindle fibers attach on this side, spindle fibers attach on this side. This is my what? Centromere. So that the in the metaphase stage, what happens? All the chromosomes, they arrange in the equatorial plane. But the difference is what? They arrange in two layers. In the mitosis, they arrange in the single layer. In the meiosis, they arrange in the double layer, right? So they arrange in the double layer. This is our what? The spindle fibers. They will go like this to the what is this one? The chromosomes. Already, that's why I'm doing very fast and I'm not giving more concentration here. And during the anaphase 1, what happens? These two chromosomes, they begins to move to the opposite pole. One goes to this pole, another goes to this pole. That's why what happens? Finally, during the anaphase 1, these chromosomes, half number of chromosomes moves to one pole, half number of chromosomes moves to another pole. Are you understanding? That's why this anaphase 1, if this pooling of the chromosomes is abnormal, then it produces what? Non-disjunction. That we discussed already. Unequal number of chromosomes, they may move to the opposite poles, right? So this is about the anaphase 1. During the telophase, what happens now? During the telophase 1, the two pole clearly get the two nucleus, but they contains half number of chromosomes, because during the anaphase 1, half number of chromosomes moves to one pole, the another half number of chromosomes moves to the another poles, right? So that what happens there, the half number of chromosomes goes up, half number of chromosomes goes down. Here the splitting of the chromosomes, the splitting of the centrosome does not occur like in the mitosis. In the mitosis anaphase, what happens? The centrosome divides, but here in the anaphase 1 of the meiosis, the centrosome does not divide. Only the number of chromosomes half move to one pole, half move to another pole, so that these two chromosomes, the diploid mother cells, it becomes haploid mother cells, N, right? So finally what happens during the telophase 1, the nuclear membrane begins to appear, nuclear membrane begins to appear, nuclear membrane begins to appear, and what happens here? The Golgi body begins to appear, the endoplasmic reticulum, they begins to appear. It happens in the telophase 1. So here what happens? Cytokinesis does not occur at this stage. Cytokinesis, after the completion of meiosis 1, there is no cytokinesis. Only the single cell membrane inside two nucleus in the two poles, both of these they contains the haploid number of chromosomes. And the meiosis 2 begins. Meiosis 2 begins. During the meiosis 2, what happens now? You know the meiosis 2? The meiosis 2 is what? Equational division. Meiosis 2 is what? Equational division. So in the equational division, what happens? We already got two daughter cells. These two daughter cells will become 2 again. 2 again. So finally they gives rise the 4 daughter cells, right? So let's go to the prophase 1. The prophase 1, same like in the mitosis. What happens here? The the chromatin begins to what is this one? Chromatin begins to condensed. Chromatin begins to condense, they begins to become short. Again nuclear membrane begins to degenerate, and you have the centromere, it divides into two and moves to the opposite pole, and they begins to show the ester formation begins. Ester formation means what? The microtubule formation begins, right? This stage is already. And in the metaphase 2, what happens? At same like the mitosis metaphase 2, they arrange in the equatorial plane. The chromosomes, they begin to arrange in the equatorial plane, and the spindles, they go and attach spindle fibers, formation completes, right? In the metaphase 2, same like in the what is this one? The mitosis metaphase, right? And then in the anaphase 2, what happens? Finally these chromosomes, they split now. They split. So half moves down, half moves up. So the number of chromosomes remain the same. Number of chromosomes remains the same. This is in anaphase 2. So in this anaphase 2 also, if this pooling is abnormal, it produces what? Non-disjunction. It produces non-disjunction. Is that okay? And the telophase 2, what happens? Finally, we have the 2 cells here, yes or no? 2 cells. Now in the telophase 2, this the nuclear membrane begins to regenerate, the Golgi body begins to regenerate, the endoplasmic reticulums, they begin to regenerate, the nucleolus, they begins to regenerate, right? So it happens in the telophase 2. After the telophase 2, only the cytokinesis occurs. Cytokinesis occurs. So at the end, what happens? We get the 4 haploid daughter cells, right? So the one in the what happens in the meiosis 2, one point you have to understand, DNA replication does not occur. DNA replication does not occur, because DNA replication, if you remember, DNA replication already occur here. This DNA replication already occur here before meiosis 1. So after the meiosis 1, during the meiosis 2, there is no DNA replication. Only the what happens? The splitting of the centrosome occurs. Centrosome splitting occurs during the meiosis 2. So please remember this one. This meiosis in humans, some points we have to remember here. So what happens in the meiosis in the human? Finally, what happens in the meiosis in the human? It creates daughter cells with 23. The 23 pairs of chromosomes will reduce to 23 number of chromosomes. In this 23 number of chromosomes, what happens? We have 22 plus 1. 22 plus 1. What is that 22 plus 1? 22 is autosome plus 1 sex chromosome. That sex chromosomes may be X or Y. Here also same, 22 plus 1, 22 plus 1, 22 plus 1. That means what? 1 is sex chromosome, that is either X or Y, that is either X or Y, X or Y. 22 are what? The autosomes, right? So that this happens in the human meiosis. Now, 2 points about the errors in the meiosis 1. If there is the error in the meiosis 1, what happens? If there is error in the meiosis 1, the homologous chromosome, they fail to separate. So that what happens? If it happens so, the daughter cell after the meiosis 1 will get more number of chromosomes. Chromosomes number will be more here, n more than n, 1 will have less than n, right? So this is at the end of the meiosis 1. So by the end of meiosis 2, if the error occurs in the meiosis 1, what happens? The homologous chromosomes, they fail to separate. So that one daughter cell will not become haploid. It will get the chromosomes number more than haploid, right? And another daughter will become less than haploid. So that as a result, what happens? It will be 4 daughter cells. So out of these 4 daughter cells, they will create 2 gametes with an extra chromosome. That will be n plus 1 extra chromosome. And the 2 gametes will be n minus 1 and n minus 1. Do you understand? Because it gets less than n chromosome, it gets more than n chromosome. So that in the daughter cells, they will divide equally again. So they will get n plus 1, n plus 1. So the 2 daughter cells will be n plus 1, 2 daughter cells will be n minus 1 if there is a problem in the meiosis 1. Meiosis 1 actually in which stage? Anaphase stage. Anaphase, that means non-disjunction occur in the anaphase 1 of meiosis 1. The final result will be what? The n plus 1, 2 gametes, n minus 1, 2 gametes. Actually, it should be what? N4 should be n, right? But if the error occur in the meiosis 1, that means the separation of the chromosomes, the homologous chromosomes should move equally. N in one pole, n should be moved to the another pole. But if the mistake occurs during this anaphase 1, these separations, then one haploid cells which we get after the meiosis 1 will have more than n number of chromosomes, another will have less than n number of chromosomes. So the result will be also the same. From this one it will come n plus 1, from this one it will come n minus 1, right? So that this n plus 1 means what? It will be trisomy. The n minus 1 will be what? It will be monosomy. Hope you understand, right? So this is the problem in the meiosis 1. Similarly, sometimes there may be the problem in the meiosis 2. So if the problem in the meiosis 2, what happens? We have here the mother cell 2n. This 2n will give you what? N and n. That means this is normal. But the problem comes over here in the meiosis 2. See, the meiosis 2 will not have a problem in both the meiosis. In one haploid will have the problem in the meiosis, right? So if the meiosis 2 have a problem, then what happens? This one which has no problem, it will produce the normal gamete as n and n. So the 2 gametes will be normal. But if this is affected, this one in the meiosis 2 is affected, then what happens? It will produce the abnormal gamete. One will be n plus 1, one will be n minus 1. That means one will be trisomy, one will be monosomy. That is disjunction, non-disjunction in the meiosis 2. So the meiosis 2 has a non-disjunction problem in the anaphase 2. It produces what? 2 normal gametes, 2 abnormal gametes. That is, two abnormal are what? One trisomy, one monosomy. But if the problem we have in the meiosis 1 has the problem, then it produces the final result what? All the gametes will have the problem. 2 will have n plus 1, 2 will have n minus 1, n minus 1. All of the gametes are abnormal. Hope you clear, right? So that we completed the meiosis. Hope you understand the meiosis. In the middle, I didn't explain, well, because it is similar like the mitosis, so that you can, get the reference from the mitosis. Only some, the major changes, I explained to you, right? So if the chiasmata or crossing over fails, then the genetic variation will be failed. There won't be the genetic variation in the daughter cells. So hope you understand about this, the meiosis. The next lecture we will try to discuss about the gametogenesis, the oogenesis and the spermatogenesis. So till then, have a good day.